All right, thank you so much for, well, coming or staying, whichever one you're doing. I'm here to tell you a bit about what we do at Synthace, where we're trying to create a digital experiment model to basically help you run better experiments. And my name is Luci, and let's get started. Drug discovery is really a long and arduous process that does take quite a lot of time, and it's very costly. And so with the recent advances in AI, it's been really exciting to apply it also to this field, where it's really helped with target identification, drug design, even predicting drug properties. But as you can imagine, there's still a lot that we can use AI for. And we can perhaps start thinking about how do we apply AI to also other parts, like helping us design better experiments. Biological datasets are used for basically generating the AI models, and this is your phenotypic and expression data, omics data, disease associations, but also published literature and patents. Like all of this is important for generating these models. But these are quite heterogeneous datasets that still need to be curated to some set of standards. So usually this is FAIR, which stands for findable, accessible, interoperable, and reusable, because it's important that the datasets we're feeding in are actually of good quality. But we need to remember that the predictions that the AI makes are still just educated guesses that need to be experimentally validated. And then hopefully we apply an iterative approach to help improve these predictions such that this data is then fed back and the models improve over time. But as you can see, there's still this step where we need to curate this data. And why? Why don't we just produce better quality data? And so this is the point where we need to start thinking about our experimental design and make sure we're creating high quality data from the outset so that we don't need to spend time curating it for that long. And that is basically what I'm trying to focus this talk on. So what is a typical experiment? So in a typical experiment, you need to obviously design and plan it, which is really the most critical part. And then you need to run it and finally analyze the data. Does this sound simple? Not quite, right? So in a usual experiment, everything resides in the mind of the scientist. They have to think about all these different components from experimental design, like how all the calculations, and if they're automating, they also need to think about all the automation instructions. Reflected in the amount of components they have to keep track of, it's also all the tools that they have to use. So they have to interact with all the different sample management, ELN systems, but likewise, there's different statistical software for design of slightly more complex experiments. And finally, once they analyze it, there's still loads of softwares to be used there. But then at the end of it, the onus is on the scientists. They need to boil down this complexity into digestible insights, into curated quality data. And so this is quite a big task. This is hard. And so I hope you can appreciate that today's challenges are really, especially if we're trying to automate experiments, across the fact that we have quite dispersed tools for each stage, which are quite inflexible to a large extent. So that means you can't really design the experiment you want to run. You need to bend to the tools you're using. And then also, if you're automating, you require significant programming expertise. But even once you succeed in running the experiment, the work is not done. You still need to spend a long time wrangling the data, getting it into that highly curated data set that you want. And so this is where we come in. We have come up with a digital experiment platform where we're trying to target all these different parts of the experimental cycle. So we're looking at design and plan, the run, and the analyze as well. And so with Synthace, you get one tool to capture the entire experiment. And your experimental planning becomes more responsive. You're no longer boxed in by the tool you're using. It's quite iterative, as I will show you in a bit. And more importantly, it's universally accessible. You don't need to be a programmer to use automation. You can just use a nice interface, and we will do it for you. And finally, because we have all this experimental context, so because we're capturing the entire cycle, we also rapidly structure your data and help you visualize it. And don't take it just from me. We also have our customers, which have found significant value in Synthace enhanced automation. So in running their experiments using us. And you might have seen this talk at last year's SLAS, where AstraZeneca presented how they could suddenly run experiments they couldn't do before. And in this particular slide, they were showing how they managed to explore a really vast experimental space using design of experiments. And what is it? How does it actually look like? So let's focus in on this design and plan stage, because what I want to show you is how you can run better experiments, so not be constrained by the tools you're using, but just really think about what is the right experiment. And so how do we communicate actions in the lab? So there's this tension between natural language, where you might write out a protocol that you're going to run, and then if you're trying to automate it, you have to boil it down to really low-level instructions for automation. And so this is generally scripting, where you're literally defining aspirate this, dispense this, and you can imagine this is quite cognitively difficult, and it's not really human readable. But we offer another way, which is really a combination of natural language, which is, well, natural to all of us, and a visual representation of protocols. And so here you can see this level of abstraction where we allow users to digitally define their workflows, their experiments, using these, what we call elements. So they're defined modes, defined actions that you would want to do during an experiment. And here you have some examples, like aliquot, dilute, make mixtures, normalize. If you think about any protocol that you run, it's really just an amalgamation of all these different actions. And these elements then have different, each element with a defined action has its own input parameters that you need to fill in. This means that it's, no matter who the user is, you have to fill in these parameters, and that way experiments get the important context captured. We then take care of boiling it down to this complex set of instructions for automation, and you don't really get to interact with that. And so you wire these elements together into what we call workflows. And so here I'm showing you a simple one where you're simply defining some liquid that you might have on a plate with some specific metadata, and then you want this liquid to be aliquoted out. And so all this will look like is these two elements strung together. And so here I'm showing you an example one, which is a Bradford protocol, where in the blue we're defining some liquids, so our standards, diluent, our samples, and then we're diluting the standard, and then we're mixing the Bradford reagent, onto the samples and standards. And so here I'm going to show you in a bit of a more dynamic fashion how it looks like in our software platform. So you see you have all these elements strung together where you simply define various inputs. And this will be, the inputs we are asking you to define will depend on the action that you've chosen. And what I haven't mentioned, we have also specialized elements for specialized protocols, such as, for example, purification. But importantly, what we then show you is a preview of what it will actually look like on automation. So I don't know how many of you have automated protocols before. You don't really get to see what the script will do, but we will show you this. So you can appreciate here in this video, which hopefully will run. You get to see every single action that will happen during that protocol. And so that means you can quickly iterate on your experimental design. You can quickly go back and change things, then simulate again, and have a preview what the robot will do in real life. Once you're happy then with this experimental design, you get to simply send this job to the lab, where you simply need to set up the deck of the robot the way Synthace told you to, and click run. That's all you have to do, and Synthace will take care of all the instructions under the hood, so you will never really have to interact with the native software. And so I hope you can see that we really captured the entire cycle where you don't get to just see one bit at a time, but you get to capture everything as a digital asset. So first off, here I'm showing you the workflow, which is immediately followed by the simulation, which shows you the preview of what will happen in the lab. And finally, because we have all the experimental context, so we know exactly what has happened during that experiment, we can allow you to easily structure and visualize the data. So you don't need to spend any time doing this manually. It's all automatically done for you. And so the main message I want to give you here is that we really accelerate your ability to get to high quality data faster. In this example, I'm showing you a workflow from CSL, who have done quite a complicated purification protocol where they were trying to examine many different parameters simultaneously. So they were running a design of experiments. And this would have been really difficult to set up via scripting, but in this way, this person who has never used automation before was able to design and execute it themselves. And so I hope you can appreciate that using Synthace, we enable more scientists to use liquid handlers. And as well as that, the liquid handlers get heavier use, so they are employed much more, which translates directly to savings on time, because we allow simulation of what will actually happen. You get to quickly iterate, you save time on programming. And more importantly, because we directly structure all the data and process it for you, you save considerable amount of time on that as well. And so here I'm showing you a direct comparison of the same experiment run via Synthace and via Tecan's native software EvoWare, where obviously you're saving time on programming the robot because it's all done via our visual interface, so you don't need to do a lot of back and forth changing. And finally, because we already have all the experimental context, so all the metadata, we also save time on data processing. But so I've shown you how we can help with the design of experiments, but let's think about how we can use AI to actually help us with the experimental design. It won't change. So going back to the slide where I talked about natural language and scripting, and I hope I showed you there's another way with Synthace. What about if there was some kind of AI assistant that actually helped and guide you? What parameters are relevant? What do you need to input? What other stuff you need to think about? And if you make mistakes, what if there was something that already corrected them for you without you even seeing the mistakes? And that's how, that is if I get to the slide, talk about a big build-up. We have decided to generate a prototype where we combine the Synthace platform with a natural language AI, which I'm sure you've all heard of, called ChatGPT, where we're trying to do exactly this, help guide the user to design their experiments. Sound?
As a proof of concept prototype, the Synthace AI interface lets a scientist describe an experiment they want to perform, then execute it on sophisticated laboratory automation equipment. Working from our office in Boston, Massachusetts, our scientist describes an experiment to the AI, in this case, a DOE-designed protocol for making growth media for T cells. Built on ChatGPT and integrated with the Synthace platform, the interface asks the scientist clarifying questions to update and refine the protocol. In the background, Synthace communicates with the AI, feeding back any protocol steps that would cause errors during the experiment. The AI silently updates the protocol to work within these new parameters and feeds this back to Synthace. With the experiment created, we can now simulate and automate it. Here we see how the robot will move in advance. Walking through the protocol step by step, our scientists can double check that everything will work how they want it to before it's sent to the lab. Here's the workflow that the AI and Synthace have created, showing the different steps needed in the experiment. Meanwhile, on the other side of the Atlantic, in Synthace's London laboratory, a colleague has prepared the robot with the necessary input plates. The machine gets to work. After a few minutes, everything is complete. Returning to the same chat window, our scientist asks the AI to scale the experiment down and run it on a different machine. With a small additional prompt, it's ready in just a few moments. Minutes later, again with everything prepared by the scientist in the lab, the same experiment runs again on a new machine. The implications of this proof of concept are significant. With AI, scientists will be able to run significantly more powerful, scalable, and reproducible experiments. All progress in the biosciences relies on experiments. Connecting them to the power of AI will be a revolution in the kind of science that we can imagine and progress we can make.
All right, so just to make it clear, this is just a prototype, so it's not commercially available yet, but just to show you that this is something we are exploring. And since I probably forgot half the things I wanted to say, please make sure you stop by at our booth, which is 117, so it's just down there. And there's these 3 guys that will help talk to you. There's another curly hair guy, which wasn't available at the time when the picture was taken, so he's also going to be there. So yeah, please make sure to stop by and have a chat, and let's see if we can work together. Thank you.